can't do your job or anything.
Hi everyone, it's great to see a good turnout both in person and online to welcome a visit from an old friend of the unit, Dave Evans from University of Queensland. Dave joined when we were called the Centre for Causal Analyses in Translational Epidemiology, which is just about the worst possible name for a centre, but we had to have translational in the title. But despite the name of Kate, as it was called, yeah, Dave came and joined us. I think it was after Dave joined us that we actually Googled Kate, and we discovered that in Gaelic it means worn out, knackered. So that was it. But anyway, then there was a program lead when we transferred to being a unit, and has worked, I think, almost as hard with people from here as one here since. Has been a great collaborator, and several of you here have visited him in Queensland, and so we're really pleased to be here for a couple of weeks to build on those collaborations.
Thanks for those really kind words, George. Yeah, I remember the Kate Centre. It was putting the casual in causal, right? Like everyone used to accidentally misspell the causal part, casual analyses in translational epidemiology. But it's great to be back in Bristol. Am I standing in the right spot for people I was told to?
Yes. Slightly, yes. You're quite tall, so slightly
back. Yeah, that's good. All right. Yeah, got the thumbs up. Yeah, look, it's a real pleasure to be back and just see how well the site is flourishing, and I really think that a big part of that actually is the respect that you engender, George, in your colleagues and your staff as well. So great to be back. So today I just want to summarize, I guess, some work from my program over the last 5 years. So that's kind of the part of the talk that's not new, I guess. So some of you might be quite familiar with some of our stuff. But I'd also like to present some new work, which I presented BGA last week, which focuses on increasing the power of locus discovery in GWAS, particularly of perinatal phenotypes. So many of you, no doubt, are very familiar with the Barker hypothesis, so-called Barker hypothesis, and the developmental origins of health and disease. I won't spend too much time explaining what that is, but basically this kind of stemmed from observational epidemiological studies looking at the correlation between things like low birth weight and increased risk of cardiometabolic disease, in particular in later life. And David Barker and colleagues kind of came up with the idea that this kind of represented some sort of environmental insult, either in utero or in the early on the life course, and then this had effects on risk of disease in later life through compensatory mechanisms and things like that. So very much kind of an environmental hypothesis. And I first got interested in this world in the context of a genetic study. Actually, I'm primarily a statistical geneticist, and only really became familiar and interested in epidemiology when I moved here in 2007. And at the time, we were conducting a large genome-wide association study of birth weight. So this was based on data from the Early Growth Genetics Consortium, so the EGG Consortium, and the first wave of the UK Biobank data. So it was published all the way back in 2016 now, which is quite some time ago. And I was interested in whether we might be able to leverage genetic information to tell us something about this purportedly environmental hypothesis. And so one of the things that we did is that we used a technique called GREML or GCTA, which basically allows you to estimate the genetic correlation and genetic covariance between traits using individual level genetics data. And what we did is we looked at the covariance between birth weight and a number of traits in the UK Biobank, including type 2 diabetes and systolic blood pressure that you see there. Might just try and get my laser pointer on, might be easier. And what we found was quite surprising. So when we did these analyses, basically what we found is that the majority of that covariance between birth weight and these cardiometabolic traits was actually due to genes rather than the environment. This was kind of a bit of a shock to us because everyone had been talking about the importance of the environment. So you can see that the coefficients there are negative. Now, we reported on this in our 2016 paper, but this table hides a subtlety. So if you look at the residual covariance, this is kind of sitting, just below zero, I guess. But this residual component is not just the environment, it's also untagged genetic variation. Okay, so if you assume that the untagged bit, which is probably more low frequency stuff, yields the same genetic covariance as the more common tagged stuff, then the implication is that that environmental correlation is actually positive. Okay, so this is kind of saying, actually, the negative correlation between the two is being very much driven by genetics. And if anything, the environment is resulting in a positive correlation. We also looked at the relationship using a method called LD score regression. So rather than using individual level data, this is where you take GWAS summary results data, and the method will allow you to get an estimate of the genetic correlation using the summary results data, which is great because you don't have to genotype the same group of individuals and measure them in all these different traits. You can use publicly available summary results data to estimate these genetic correlations. And if you can see on this plot here, RG is your genetic correlation on the y-axis. This is with birth weight. And then on the x-axis are all these other traits that have been grouped according to their particular domain. So if you kind of look in the cardiometabolic area, you can see that that genetic correlation is kind of very much negative. So again, that's saying that the source of this correlation, there's at least a substantial kind of genetic component there. But again, there's a subtlety, because when you're dealing with a perinatal trait like birth weight, that trait's not just the result of your own genetics, but it's a result of maternal genetics as well. And so you could envisage a situation that, sure, there's a genetic correlation, but it's actually being driven by mum's genome that then impacts the intrauterine environment that then has an effect on birth weight and other perinatal traits. And so what we were interested in this next paper was to try and dissect those two things, the maternal contribution and the fetal contribution. So this is work that was led by an incredible postdoc of mine, Nicole Warrington. What you see in this Miami plot here is on the top bit is GWAS of own birth weight. And on the bottom, which is almost but not quite a mirror image, is GWAS of offspring birth weight. And I guess the point here is that both those GWAS represents a mixture of maternal and fetal genetic effects, okay? Because obviously your own genome is correlated with your mum's genome and vice versa. And so we were interested in trying to disentangle these two things. I should say that this is predominantly in UK Biobank and also the EGG Consortium, and these are unrelated individuals. So how would you disentangle those contributions normally? Well, if you're lucky enough to have mother-offspring pairs, you just do a conditional analysis, right? So you take birth weight and you throw in your own genotype and your mum's genotype, and you just perform a conditional regression analysis. I've also pruned down structural equation model in this figure because I like pictures rather than equations. And this will kind of give you an idea pictorially of what's going on. So you've got birth weight and you've got influences through both the maternal and the fetal genome, and these things are correlated because of the transmission there. So you could fit this model using SEM or just by regression, you'd get the same answer. The problem, of course, is that there's not many cohorts in the world that have these data. So here in Bristol, we're lucky enough to have one of those. And the Norwegians have a lot of these data as well. But in general, there really is a paucity of these mother-offspring pairs. And so we were interested in whether we could use unrelated individuals to leverage the power of UK Biobank. Because UK Biobank is unusual when it comes to birth weight, because you have people self-reporting not only their own birth weight, but they also self-report, in the case of the women, the birth weight of their first offspring. And so we realized that you could use a structural equation model to get an estimate of maternal and fetal effects on birth weight and partition those genetic effects into maternal and fetal components. So here's again an SEM, pruned down SEM of how we do that. So birth weight, your own birth weight of your offspring, you've got your genotype here. And of course, you know that the individual's mother who haven't genotyped is going to be correlated 0.5 with the observed genotype, and likewise, the genotype of their offspring is also going to be correlated 0.5. And so basically you can fit this model and get estimates on those parameters and do this in very large samples. So we published on that in 2018. Again, this is work led by Nicole. And this is what we found applying that method to the known loci for birth weight at that time in the UK Biobank. So on the y-axis is the estimate of the maternal genetic effect. On the x-axis is the estimate of the fetal genetic effect. Note that the scales are different. So the fetal effects are bigger in general than the maternal effects. So you can see that in most instances, most loci have some sort of fetal effect, but there's a few that have predominantly maternal effects. And then interestingly enough, there's a cluster of loci that have both effects in discordant directions. So when they're present in the mums, they do something to birth weight. When they're transmitted to the kids, they do the opposite to birth weight. And interestingly, a lot of these are known type 2 diabetes variants, and that actually makes sense if you know something about the biology. So basically, these things do things like increasing fasting glucose when they're present in mums. So the baby senses the glucose, grows more, but when the risk alleles are transmitted to the offspring, it means that the babies can't use that glucose as well. They're kind of more insensitive to it. So it makes sense also from biological perspective. So we took those estimates for maternal and fetal effects right across the genome, and we did that LD score regression analysis where we estimate the genetic correlation between birth weight and all these different traits. But this time we have a fetal component and we have a maternal component. And This is not a perfect sort of analysis, but I think it was quite an interesting thing to do. And I'll draw your attention to the red circle again. So the blue ones in the circles here, that's basically correlating with the fetal effect, whereas the red triangles are correlating with the maternal effect. And so it's kind of suggesting positive correlations with birth weight through the maternal genome, negative correlations through the fetal genome. There's some other sort of interesting things here in this diagram as well. Educational attainment and IQ. Doesn't look like there's any fetal effects, but it looks like there is a positive correlation with maternal IQ, which again is kind of what you would expect intuitively. Okay, so that analysis that I showed is not perfect for a number of different reasons. One of them is that although we've kind of partitioned the genetic effect on birth weight into a maternal and a fetal component, we didn't partition the other traits into maternal and fetal components. And so this is a paper that if you've been following this work, you may not have seen. So it was published earlier this year in Behavior Genetics, and it was led by Helen Moon, who many of you may know. And the idea here was, I might just show the scary SEM, is to partition that genetic covariance between birth weight and cardiometabolic traits into maternal and fetal components. So you've got that partitioning for birth weight, and now you've got it for the cardiometabolic trait as well. And so all this was done in a software called Genomic SEM. I'm not going to show you any empirical results because we did this in the Hunt cohort together with UK Biobank, and even with the tens of thousands of folks in Hunt, we still didn't have a big enough sample size, I think, to have meaningful results that I believe, basically. But I think the method is totally sound, though, so it's primarily kind of interest with respect to that. So that's looking right across the genome using LD score techniques and GREML techniques. But we can also obviously utilize single SNPs to investigate this. Well, I'm not going to go over MR because you guys are obviously all experts in that, except to say that when you are looking at maternal environmental exposures and offspring traits, it's very important to consider a possible pleiotropic path through the offspring genome. So obviously mums transmit their SNPs to their offspring, and so this pathway could be having an effect on offspring outcome. And so it's really important to block that path by conditioning on offspring genotype. So that's easy to do if you've got mother-offspring pairs. But you can also do this in a two-sample context as well. So if you have estimates of maternal effects on offspring outcomes and fetal effects on offspring outcomes, you can use those estimates in a two-sample MR kind of framework. And if you're interested in sort of reading how that can be done, take a look at our paper in 2019 in the IJE. So here's one example of doing that. So this is just looking at fasting glucose on offspring birth weight. So the red rows are when you don't condition on offspring genotype. And so you can kind of see here that there is a causal effect from the mothers, a positive one, not much happening from the fetal genome, but the picture looks a little bit different if you then basically adjust for offspring genotypes. And now that maternal effect becomes even stronger, but you can see that there's something going on from the fetal genome as well. So that's looking at kind of maternal effects on perinatal traits, but we're obviously also interested in later life traits like cardiometabolic disease. And so we sort of came up with this framework to try and answer some of those interesting Barker hypothesis and DOHAD-like questions using an MR framework. So again, this is work that was led by Helen Moen. And so the idea here is that you can take maternal SNPs for exposures that you're interested in. So it could be something like maternal blood pressure or fasting glucose or something. In Helen's study, she looked specifically at birth weight associated SNPs in the mum. So basically maternal SNPs that are known to affect offspring birth weight. And we reasoned that it was informative to look at those SNPs and see then whether they were also related with risk of cardiometabolic disease in the offspring. And we reasoned that there were probably 4 different ways that this could happen, according to this panel. So the first way was that the SNPs could affect the intrauterine environment, which would decrease your offspring birth weight. But then it would result in this kind of cascade that would eventually lead to increased risk of cardiometabolic disease. So this is very much kind of consistent with Barker and DOHAD and that sort of thing. In panel B, you can have the same sort of thing, but it's actually a direct consequence of an effect on offspring birth weight. I don't think many people would think that this is exactly what happens, but it's possible. This could also be a possibility. So again, kind of DOHAD, Barker consistent. Panel C is something very different. So here it's your maternal SNPs are related to offspring birth weight, but the reason why they relate to offspring cardiometabolic disease is they're transmitted to the offspring, and then these variants then pleiotropically affect risk of offspring cardiometabolic disease. So this is absolutely not sort of consistent with Barker-like mechanisms. And then, of course, there's also the possibility that there could be some postnatal effect of those maternal SNPs. But if you're fortunate to have father-child dyads in your sample, you can test this as well, because you would perhaps, if it's a postnatal effect, you'd perhaps expect to see it in the fathers as well. So Helen had a look at this in the Hunt cohort, specifically using birth weight SNPs as her instrument and looking at all these different outcomes. And kind of the take-home message at the end of the day is that there was no association or no significant association between maternal or paternal GRS and these offspring phenotypes after conditioning on offspring genetic risk score as well. But the reverse wasn't true. There was actually quite substantial correlations between offspring GRS and offspring phenotype after conditioning on maternal and paternal GRS. And so it seems that for these birth weight variants, again, what's driving that correlation is really the fetal genome. It's not through intrauterine pathways. Oh, that's a photo of Helen as well for folks who don't know her. So we've done a number of other studies as well using this sort of paradigm to investigate maternal exposures and offspring risk of disease. Here's another one, a PhD student of mine who's just graduated, actually, Gang Wang. He looked at systolic blood pressure and future risk of cardiometabolic disease, again, in the Hunt cohort. And again, the bottom line is it very much looks like the observed correlations are driven by pleiotropy in the offspring genome. So in this world, a key kind of limitation is power. So I feel like the studies that I've shown, we've been able to kind of rule out larger genetic effects, but we've not been able to rule out smaller genetic effects. So this is the maternal and offspring genetic effects power calculator. This is something that Helen did in her PhD. So if you log on to this site, you can get estimates for the power of your sort of genetic association studies in this kind of world, and it's rather useful. But the power calculations were quite sobering in that, you know, given the amount of mother-offspring pairs around the world, you know, we didn't have great amounts of power. And so we had to think about ways in which we can improve that situation. And so one of the ideas we came up with was to impute parental genotypes if you've got other relative pairs in your study or your biobank. So the idea here is if you have something like a SIB pair on the left here, you know, if they're AA, BB, then you know that the parents are going to be AB, AB, right? And so the bottom line is if you have related individuals like SIB pairs and half SIB pairs, you can impute parental genotypes and then use those genotypes in your downstream MR analyses and genetic association analyses. There's some limitations in doing this. So, for example, from sibling pairs, you can't tell maternal and paternal genotypes. You can kind of only resolve an average parental genotype. But if you're looking at parental effects, you can kind of argue that dads don't matter or dads only have a small effect. And so that's perhaps not a massive assumption in this world. So this was published back in 2020, and we did release some software for that called IMPISH. So IMPISH is super easy to use. So, you know, you can do this analysis easy in half a day. The field has moved on since. So a guy called Alex Young has published a method, implemented a program called Sniper. So Sniper IMPISH uses just identity by state information. So it just looks one SNP at a time and does the imputation there, whereas Sniper looks at haplotypes basically and uses identity by descent information. So Sniper is much more powerful. The problem I think at the moment is that we at least have found it extremely difficult to use. It's not particularly user-friendly. So something to bear in mind if you're in that world. The other thing that we have been doing, of course, to increase power is rather than just depend on cohorts with mother-offspring pairs, is we've been developing approaches to utilize summary results data. You have maternal GWAS and fetal GWAS. So here's one example of that. Again, this is led by Nick. And it uses genomic SEM to basically you take a GWAS of own birth weight, a GWAS of offspring birth weight, you fit this model in genomic SEM, and it will give you estimates of maternal and fetal genetic effects, and it will account for any cryptic relatedness between the two scans, whether you've got overlapping individuals, whether you've got related individuals, and that will kind of do that for you. The problem, I guess, is that genomic SEM is also not particularly user-friendly. And so there's a little bit of a steep learning curve here, even though this will work. And so this kind of framework, you could actually use it for everything that I'm about to present. It's just, you know, there's a learning curve and the software is not particularly user-friendly. Whereas what I am going to present is super user-friendly and very, very quick. And so it's software that we've called DINGO, Direct and Indirect Effects Analysis of Genetic Loci. I like the name because of the Australian connotations, although talking about dingoes and babies is maybe not a wonderful thing to do. Unfortunately, since we coded this software, I've got a dog myself called Odin, and I was thinking, well, that's got a D for direct and an I for indirect, and now I'm kind of regretting should have called it Odin instead of dingo, but there we are. So I might just kind of skip that. So dingo is very much about increasing your power to detect loci in GWAS. Okay, so in the egg consortium and the perinatal world, the way that things are done currently is you do separate GWAS. You have a GWAS of own birth weight, where it's predominantly kind of fetal, and you've got a GWAS of offspring birth weight, where it's predominantly kind of maternal. And then, you know, you sort of report on both GWAS, and you might follow up with some conditional analyses to work out what's a maternal locus, what's a fetal locus, what's doing both. But it's not statistically optimal because, you know, there's information that you can combine from both GWAS together, and that's, people are just not kind of doing that, in part because there's this question of cryptic relatedness across the scans, and it's difficult to know how many, you know, what your effective N is in both samples. And also in this world, people don't sort of seem to worry about the increased statistical penalty due to multiple testing and all these different things. So a little bit of maths here, not too complicated. So if you just take the ordinary least squares estimates by regressing birth weight or offspring birth weight on your own SNP, that will give you inconsistent estimates of maternal and fetal effects, okay? So if you take the OLS estimate from the fetal GWAS, you can kind of show that that's actually not the true fetal effect given by this Greek letter here, but rather it's that true effect plus half the maternal contribution. And likewise, your maternal OLS estimates are also inconsistent as well. And getting consistent estimates is important to determine whether the effect is through the offspring or the maternal genome, but it's also obviously important for downstream Mendelian randomization analyses because you want to have consistent estimates of your genetic effects. So it's very easy to do that. You can just take a weighted linear combination of estimates from the maternal and the fetal scans and weight them by 4/3 and 2/3. And when you kind of do the asymptotics, it works out so that you now have consistent and unbiased estimates of your maternal and fetal GWAS. So it's just a simple kind of weighted linear model. You can get estimates of the sampling variance for each of those estimates and covariance as well, just by doing standard kind of covariance algebra. The tricky bit is that you've got these terms here, these covariance terms, and these covariance terms arise because of the, you know, this cryptic relatedness between your maternal and your fetal GWAS. But you can actually estimate those covariance terms using bivariate LD score regression. And it turns out that the sampling covariance is a function of that bivariate LD score intercept. So it's quite a simple little formula here. So knowing that is going to allow you to combine information from maternal and fetal GWAS, taking into account sample overlap. Like so. So that's kind of the expressions in green. So very, very, very easy and fast to calculate. So what can you do with this? Well, first of all, you can do 1 degree of freedom conditional tests. So this is just, you know, does this locus operate through maternal or fetal genomes? And so that's just taking your estimate for the effect over its standard error, and it's a normal, you can test it against a standard normal distribution in large samples. The problem is that these conditional tests, they're good for locus characterization, but they're not good for locus detection. So the power is not very strong. but they're of course good for downstream 2-sample MR studies. So what we were interested in is whether we could kind of optimize that power for Locus Discovery. So the two things we did, the first was we constructed a 2-degree of freedom test. So now we're taking that fetal and maternal estimate together and we're modeling it in terms of a bivariate normal distribution. And so by some pretty standard maths, you can formulate that model into a 2-degree of freedom chi-square test, which is basically your estimates and their transpose multiplied together using the inverse of the estimated covariance matrix between the two. So this will give you a chi-squared 2-degree of freedom test of what is there a locus influencing birth weight at this position in the genome. The other thing we looked at was something even more simple, and that's just doing a 1-degree of freedom meta-analysis. I'll illustrate that using just fetal genetic effects, but we also do this for maternal genetic effects as well. So basically, you've got your fetal GWAS, your GWAS of own birth weight, and you take your estimate from it. Happy days. But you've also got your GWAS of offspring birth weight. So this is kind of like your maternal GWAS. So all you do is you take twice the estimated regression coefficient from there. You then put these two things together in an inverse variance weighted meta-analysis, taking into account your estimated sample overlap, and you get an overall estimate of your fetal genetic effect. Okay, and you just evaluate it against your standard normal distribution. So you do this for your fetal effect, and you also do it for a maternal effect, and you look to see whether either of those is significant at your locus. Now, the estimates that you get out using this are biased, they are inconsistent, all the rest of it, but that doesn't matter in this world because you're just interested in whether there's a signal at that locus. You don't care, you're not trying to estimate the effect, you just care, is there a signal or not? And the type 1 error rate and the coverage is kosher under this model, even though it's biased and inconsistent. So this is what the results of some simulations look like, comparing the usual strategy of just doing a separate maternal and fetal GWAS and seeing, is there a signal in either of those scans at a locus, the 2-degree of freedom test, and this 1-degree of freedom meta-analysis. So I'm not sure I can get rid of that annoying thing at the top. There we go, magic. Okay, the top row is when there's no overlap, the extreme situation where there's no overlap between maternal and fetal GWAS. The bottom is where there's complete overlap. The column on the left is when the locus has a maternal and offspring genetic effect in the same direction at the locus. The one on the right is where there's a maternal and a fetal effect at the locus, but they're in opposite directions. And then the one in the middle is when there's a maternal effect only. The 3 lines are, the blue one is where it's a 2-degree of freedom test. The red one is the 1-degree of freedom meta-analysis, and the orange one is the traditional strategy. So when there is a concordant maternal and fetal effect, 2-degree of freedom test and 1-degree of freedom meta-analysis, they do very well, and they're about equivalent. Much better than the usual approach, particularly in situations where you've got no or only partial overlap, which is probably most of the situations we have in the EGG consortium. Looks quite interesting when you've got loci of discordant effects here. The 2-degree of freedom analysis really shines through. The 1-degree of freedom thing bombs out really badly. So this is kind of interesting because in the perinatal world, it is not uncommon to have loci that have discordant maternal and fetal effects, as we've seen in the previous slides, right? So that was interesting. And then when you only have a maternal effect or only have a fetal effect, which approach is best depends on the degree of sample overlap. So when you have lots of sample overlap, you're probably just better off doing separate GWAS because you're not getting extra information from the different GWAS. However, when you've got no overlap or only partial overlap, the 1 degree of freedom meta-analysis is probably best. So how am I going for time?
You've got 14 minutes left.
14 minutes, okay, happy days. So this is what it looks like applying those approaches in empirical data. So this is the latest GWAS of birthweight. So this was run by the DCODE group, combining the DCODE cohort with the Warrington et al. Paper. And so that's what you see in the top panel here. So I think under the little Zoom banner there, it's like 260 SNPs or something like that, independent SNPs reaching genome-wide significance. If we take those data and use the 2 degree of freedom test, our number of independent GWAS significant SNPs, increases substantially by about 70 or so. When we use the 1 degree of freedom meta-analysis, we actually do better than that. So we're up to about 369, although notably the 1 degree of freedom meta-analysis doesn't capture some of the loci captured in the 2 degree of freedom test. And they are those loci where you have effects in opposite directions. This is all very much work in progress, but the results are pretty stark. This is just one example of a locus that has effects in different directions. I won't go into that in great detail. So this is Dingo. Basically, it will implement all the different tests that I've been talking about today. It will do it super fast, and it will account for all sorts of sample overlap and cryptic relatedness in your data sets. It's implemented in something called the Complex Trait Genetics Virtual Laboratory. So this is a platform that doesn't just do Dingo, it does loads of other things from LD score regression to GWAS and all sorts of bioinformatic analyses. So take-home message, Dingo is a simple and fast method to increase the power of locus discovery, particularly for traits that are influenced by direct and indirect effects. You can use it just using GWAS summary statistics. It gives you a range of different tests. It will also give you conditional estimates to use in downstream 2-sample MR studies. What I haven't presented today is that we've extended all the methods to be able to include paternal effects, if that's something that you want to look at. And we've also done it for binary traits as well. So everything I've presented so far is assuming nice quantitative traits, but you can also use a similar sort of framework for binary traits as well. We also have a series of power calculators as well that will enable you to calculate power for all sorts of different study designs using Dingo and other of our software. Do I have time to quickly chat about 1 or 2 unrelated things, or do you want me to finish up very quickly? Okay, very, very quickly. Something else that we've kind of looked, not related to DOHAD necessarily, but in this kind of world, lots of people who report being adopted in the UK Biobank. We were interested, can we leverage this knowledge in any way? And so we thought, well, one thing you could potentially use that information to do was to partition maternal effects into pre and postnatal effects. And so cutting a long story short, we came up with a structural equation model in order to do that. So if you're interested, have a read of Daniel's paper in eLife. The bottom line is that I suspect that people in the UK Biobank, when they say they're adopted, they're actually adopted by their relatives. And if that's the case, if you're adopted by a genetically related individual, this model's not going to work because you're making an assumption that they are unrelated. So it would be wonderful if Biobank could give us information on, you know, whether you're adopted by somebody related to you or not. But anyway. And another thing we've been doing is there is an effect at the major histocompatibility locus on birth weight. And so we were kind of interested in using our SEM dark arts to try and decompose that into maternal and fetal effects. So this is the beast of the SEM. It's even worse than that because these little ellipses, the plural of ellipsis, in the left basically means that you can repeat that structure, you know, tens of times on the other side of that slide. But anyway, that will give you estimates of maternal and fetal genetic effects from the HLA on offspring birth weight, and that is back at the IJE under consideration. And so I just want to end by particularly acknowledging the different cohorts that have contributed to this, that totally would not have been possible. This work would not have been possible without them, including ALSPAC, Biobank, and particularly the Norwegian guys, the different funders, and my amazing group. You'll probably recognize a few folks here. So John, who actually got his start in the IU back in 2009. He was a PhD student with myself and John Tobias. John's doing amazingly, got his own group now and his own position and on to bigger and better things. Nicole Warrington as well, who's does a lot of collaborative work with Kate Tilling and others at the unit. Helen Moen, who many of you will know, Daniel Gung, and all the other folks in the Evans lab. Thank you very much.
You can see why Dave had to leave Bristol though, because he's got a can of Diet Coca-Cola on the table when everyone else is having a good time. Any questions?
So you've obviously focused on birth weight for obvious reasons, but are there other traits where you think this is really, really going to be valuable?
Yeah, no, absolutely. So great question. We focused on birth weight because there's the most data for birth weight and we're ensconced in that world and it's easy for us to do. But there's loads of other perinatal stuff that we are in the process of using the same approaches for. So things like gestational age, placental weight, but you could actually use the same sort of model to look at phenotypes like preeclampsia, gestational diabetes. So now you're flipping the model on its head, but it's exactly the same principles. So the perinatal stuff, we're very much doing that, and we think it's preliminary analyses we think show us very, very useful. I guess the big question is whether, to what extent you can do it for later life stuff. So, socially patterned things, educational attainment is an obvious one. Problem is, at least in our model there, we don't have paternal effects, although you can easily put them in. But then you've got to grapple with issues due to assortment, which is a little bit more tricky. So I think, yes, it can totally be done, but we haven't looked specifically at that yet.
So once you've used the bingo method to identify locus, it's come up this year, but then is the idea that you then go back and use method is the unbiased coefficient
Yep.
to identify whether it's
Yep.
from the mum or the offspring.
Absolutely. That's exactly what we do.
I mean, there was, fashion for looking at variance across, variants of trait across SNPs, the thing
Yeah.
that they might be aware of uncovering processes like imprinting,
Yeah.
parental origin effects, etc.
Yeah.
With the quite a lot of the, it's very striking that quite a lot of, quite a lot of these SNPs are showing effects in opposite directions from the maternal,
Mm.
and that's going to do very strange things to the virus, isn't it? It's a way of, is there a way of actually then just using the variances to identify those?
Yeah, so I think that's a really interesting idea. I think you're right that there is additional information that we're not capturing through effects on the variants. And yes, you probably could incorporate that in some way. I mean, as you know, tests that rely on variance differences tend to be quite underpowered. But I think you're right that it could probably increase the power of what we are doing further. I don't know whether you sort of saw it, but you must have, you might be on the paper actually. So Charles Loren, who worked here for a little while, we tweaked the GREML model to put imprinting into, so you could, kind of on the list to do is to use that model in MOBA or something like that to get estimates of imprinting. What's really interesting though is that, and I didn't realize this, Loic Yengo had to show me this, but you actually don't need phased information to detect imprinting because it results in sort of variance differences, as you say. So you can actually pick it up in unrelated individuals. I mean, you need to make assumptions as to what is causing those variance differences, but and you need very, very, very large samples.
Yeah. But I think it would be worth thinking through. I think that you might be able to do the same.
Yeah, I think you're right. I think you're right.
The other? Dave's around for another week and a half.
Yeah.
And are you sitting in Lindsay's office all the time?
Yeah, I'm in my old student office. Very excited about that.
And we have the walls padded. It's a very good walls, Dave, and everything. We've got everyone here.
I miss hearing the arches every day, actually, George. You had the office next to mine.
The pandemic cured me of the arches. Given it up after 40 something years. Anyway, I'm really great seminar, and I managed to see Dave while he's here. Thanks, Ben. Take that slide.
Yeah.